based on what you put in your interactive notebook, what do you think we're going to talk about today? What was that, Lewis? The differences between mitosis and meiosis. Okay? The differences between mitosis and meiosis. So, let's come up with an objective. What do you think an objective of this would be? One of our objectives. Who can state one for me? An objective. Not Lewis. He's already answered a question. Somebody else. Something that we're going to do. All right. Fletcher, what's an objective? Identify key differences between mitosis and meiosis. Okay. Okay, excellent part. Okay, that's probably the biggest objective today. What we're going to learn, though, is, are there similarities? Yes. Are there differences? Are there things unique to meiosis? Yes. Are there things unique to mitosis? Yes. Are there things that both of them have together? Absolutely. So, our goal is that right there, our objective. To be able to say, okay, this is meiosis. This is only mitosis. These are both. Now, do you think that's going to come in handy when we do our homework, which is a Venn diagram? You know what a Venn diagram is, right? We've done Venn diagrams in here. Do you think this stuff's going to come in handy doing that Venn diagram? Yeah. Yeah, it is. Okay? So, let's get into some of the juice of the whole idea. Meiosis. Key differences from mitosis. First of all, meiosis reduces the number of chromosomes by half. Reduces the number of chromosomes by half. Now, if I were to ask you to use some scientific terms to explain this, what would you say? What would you say? Any ideas? This is probably a good objective to have. Reduces from half. What are some scientific terms that we could use? Okay, yeah, it's dividing, but, I'm thinking of two terms that we use to mean, one of them meant half. Huh? Haploid, right? Haploid. What was the one that didn't mean half? Diploid. Okay? Remember I said, haploid begins with an H. Haploid, half. Diploid begins with a D. Diploid is double. Okay? So, haploid, diploid. So, if we're talking about meiosis here and how it reduces the number, so meiosis reduces the number from a diploid to a haploid. Reduces the number of chromosomes in half. Daughter cells differ from parent and from each other. The daughter cells differ from the parent and each other. I want you to flip back to page 12. I want you to look at those 4 daughter cells that we drew. Okay? I'm talking telophase 2, where you drew 4 different cells. Are any of those cells the same? Any of those cells that you drew the same? What do you think, Sydney? No. Okay? None of them are the same. They each have 2 chromatid in there, don't they? But are the chromatid the same? No. Okay? What's the big reason? Here's a $10 question. What's the big reason that those chromatid are not the same? Crossing over. Excellent, Lewis. Crossing over. And Jonathan, when does crossing over occur? Prophase number 1. Those are some reviewing some of our old objectives, right? That's important stuff with meiosis here to know. Okay? Oh my goodness, it seems like we're remembering stuff. That's just like, it just meant it was big, okay? Just meant it was big. I didn't actually mean us paying out $10. What, you think I'm rich? I'm rich on life, man. All right, so reduces the number in half, okay, diploid to haploid, okay? Daughter cells are different than the parent cells. In mitosis, they were identical, weren't they? In mitosis, they were identical. The same cell was duplicated. Meiosis involves 2 divisions. Mitosis only involves 1 division. Mitosis is 2 divisions. Excuse me, check. Meiosis is 2 divisions. Mitosis is 1 division. Okay? How do you remember which one has 2 divisions and which one has 1 division? Well, didn't we talk about meiosis 1 and meiosis 2? The stages of meiosis 1, the stages of meiosis 2. Didn't we talk about those? Well, then meiosis has 2 different divisions. Okay, has 2 different divisions. Now we're looking specifically, excuse me, specifically at meiosis 1. Some differences of meiosis 1. Synapsis. Okay? Synapsis. What synapsis means is that the homologous chromosomes pair up. Okay? The homologous chromosomes pair up. Now, once again, if I were showing you this, remember when I drew the 2 circles as so, and I said, let's look at metaphase. Okay? Let's look at metaphase. Metaphase in mitosis. Okay? We have large red chromosomes and small red chromosomes. We have large blue chromosomes and a small blue chromosome. But when we were in mitosis, they all lined up on the equator as so, right? Now, when we were talking about meiosis, okay, when we're talking about meiosis, when we get to that metaphase 1, the big X is matched up with the big blue X is with the big red X. The little blue X is matched up with the little red X. So, the homologous pairs, and that's what, get a different color, that's what these are. These are the homologous chromosomes. The homologous chromosomes are matched up. Okay? The homologous chromosomes are matched up, not, okay, not matching up just the sister chromatids as we do in mitosis. Okay? Can we understand? Do we understand the difference in these two? Can you see those there and how it's lining up? I think a visual helps us a little bit. Okay, does that visual make sense? Would you be able to identify, A and B and be able to say, hey, this is A and this is B, and be able to say, okay, what stages are these? Okay? Because we're going to need to know that. We're going to need to be able to do that. All right, let me get this off of my slide because I'm going to fill up other things on the slide. So I get this off. So one thing, synapses. Second thing, we knew this one. We already talked about this one today. Crossing over occurs in prophase 1. Do we have any crossing over happening during mitosis? Do we have a prophase 1 in mitosis? No. Okay. Crossing over, prophase 1 has to be meiosis. We talked about this one. Metaphase 1, the homologous pairs. The homologous pairs lining up at the metaphase plate. Okay? In metaphase 1, the homologous pairs line up, not the sister chromatid. The final thing, in anaphase 1, sister chromatids do not separate. Okay? Both of the sisters go to the same pole. Okay? Both of the sisters go to the same pole. And if you remember, okay, way back, let's turn back to 8. Let's turn back to page 8 in your book. Okay? It's tough. It's even tough for me to remember, folks. But we need to be able to tell what sister chromatids are compared to homologous chromosomes. Okay? Sister chromatids compared to homologous chromosomes. So, if we look and we draw it again, let me get a little space here to draw these, all right? So if I were going to draw these again, all right, we said that if I would have just one chromosome, it could look like this. If we draw that chromosome duplicated, we would have a centromere, and then we would get the X-looking guy, right? So that right there is the sister chromatid. The sister chromatid right there, because what are the sisters? Here's one sister. Here's two sisters. Now, could I have a green one? Duplicated as so. Let me get that bottom. Okay. Could I have a green chromatid that was duplicated like this too? Would those be sisters? Yeah. These would be sister chromatid. Now, the ones, okay? The blue one and the green one, when we pair these two up, we're going to call them what? Homologous chromosome or homologous pair. Okay? A homologous chromosome or homologous pair. So that's just a little bit of a review. And that on page 9 is important to know the difference, because you can see that in meiosis and mitosis, whether they're lined up as homologous chromosomes or whether they're lined up as sister chromatids, it makes a difference, doesn't it? Okay? It makes a difference. All right? So,